is a private citizen, so he can say whatever he wants as of August of this year. But for almost the entirety of his career, he was at NICHD, leading a branch as a branch chief. But we came to know him as one of the strongest advocates and partners in vision for CBTN. And as Roland mentioned, for the potential of leveraging a common framework on behalf of larger scale impact in a global fashion. In 2017, this is only a few years after CBTN was started, we had the opportunity to become part of the Kids First program, which James will talk about. It was a tremendous leap of faith by NIH at the time to invest in an unknown entity to become part of a national infrastructure building initiative. And it's really a credit to NIH as a true partner in vision, especially at NICHD at the time, to elevate and not only address the unmet need of pediatric brain tumors and pediatric diseases writ large, but to recognize, just like Roland mentioned and AWS has recognized, that our community is actually a hub of innovation to create new solutions writ large. And that's an inversion of the opportunity context that James will highlight as part of his talk next.
All right. So, good morning. My name's James Coulombe, and until I retired at the end of August, I served as the chief of the Developmental Biology and Congenital Anomalies branch at the Eunice Kennedy Shriver National Institute of Child Health and Human Development, which is exhausting just to say. But I'm here today. I'm not a cancer biologist. I'm an embryologist. But I'm here today because, in addition, it had been my honor to serve as the NIH Working Group Coordinator for the Gabriella Miller Kids First Pediatric Research Program. And Adam asked me to give this little introduction because I was involved with that program from the very early stages and can probably tell you more about what was behind the scenes than anyone else. So Kids First was started in response to the 2014 Gabriella Miller Kids First Research Act. The act and this program were named in honor of Gabriella Miller, a young Northern Virginia girl who passed away at the age of 10 from diffuse intrinsic pontine glioma. But in her short life, she became a very persuasive advocate for pediatric cancer research. The act authorized an appropriation of $12.6 million per year for 10 years to be used to support pediatric research, specifically to supplement, not supplant, other existing NIH research efforts. Kids First has been a trans-NIH common fund program administered by the NIH Office of the Director and NICHD with the help of colleagues at NCI, NHLBI, NHGRI, and staff from several other institutes across NIH as well as the CDC. From the start, the emphasis has been on sharing genomic data, phenotypes, and clinical information, and making these data available to the broad community of researchers and patients. Kids First is focused on birth defects and cancer because these are leading causes of childhood mortality. It also makes sense to study these conditions together since shared genomic variants and signaling pathways have been identified, and a child born with a birth defect faces a higher risk of also having cancer. As a developmental biologist, it's fairly obvious that cancer is a problem of growth control, which is also part of development. And so the two are very linked. Although collectively, these conditions are far too common, individual conditions are mostly rare, and collecting sufficient patient populations for genomic studies has been challenging. But Kids First was conceived to surmount this problem by building a data resource of high-quality genomic data from affected children and their parents, harmonized by mapping against the same reference genome with variants called through the same analysis workflow. And this data resource was explicitly intended to be shared so that data from studies of a given condition or shared phenotypic or clinical aspect across conditions could be combined. And 10 years ago, that was cutting edge. That was supplementing, not supplanting anything anyone else had conceived of. With the first appropriation of funding in 2015, we began our efforts to build the data resource for Kids First using a somewhat unusual NIH mechanism that's called an XO1. And the way it works is that investigators respond to a notice of funding opportunity to apply for sequencing services. These applications are then peer-reviewed by a special emphasis panel organized by our colleagues at NHGRI. The Kids First Working Group then prioritizes the requested services according to impact scores and specifically according to program priorities. After samples are provided to our funded sequencing centers and after the genomic data is generated by our funding sequencing centers, which are at the Broad and Hudson Alpha, the Kids First Data Resource Center aligns that data with phenotypic and clinical data provided by the investigators and harmonizes genomic data. And once this surprisingly long process is done, the genomic data is provided to the investigators for a period of 6 months of exclusive access before it is then made publicly available through the Kids First Data Portal and NIH's dbGaP. But it's one thing to assemble a trove of data and provide a few investigators with data from their cohorts. To make the data more valuable, it is important to provide broader access in ways that fit the needs of the user community. Shown here is the landing page of the Kids First Data Resource Center. This effort is organized out of the Children's Hospital of Philadelphia, but it's actually a collaborative effort of investigators from several institutions across the US and includes contributors from Canada, Italy, and even Serbia. In addition to acting as a data coordinating center, an important aim of the Data Resource Center is to facilitate user-friendly access to both summary and individual level data for clinicians and basic scientists, and to also provide data analysis tools for use in the Cavatica cloud-based analysis platform. So the Data Resource Center has built a portal where users can visualize clinical and phenotypic data, filter the participant files, and even construct synthetic cohorts combining individual genomic data across studies and conditions. And accessing the data is really quite straightforward. A simple registration, which just takes a few minutes, and a click-through agreement and login allows a user access to the descriptive, phenotypic, and clinical data. This allows users to formulate hypotheses and determine the feasibility of analytical projects before requesting dbGaP access to the individual genomic data sets. That's also something that's kind of unique to Kids First because mostly the data is walled behind dbGaP access requests for NIH facilities, and you can't dive into the data first to generate your hypotheses. You got to get the whole access to the data set, know what you're going to do first before you can get that. So Kids First has been unique in that. But in keeping with NIH genomic data sharing policy and consent agreements with the contributing patients, the genomic data is made available through dbGaP access requests. To facilitate this, minimize potential delays, we formed a Kids First specific data access committee. And although it says here something like that approvals generally within a week, in practice, it's often as short as 1 day. So we've really tried to speed up what has been viewed as a barrier to access. With access to those genomic files, a user can then use the Cavatica cloud-based platform to bring Kids First data along with data from other interoperable NIH data efforts or from their own data into an individualized workspace. They can also bring authorized collaborators into their Cavatica workspaces, and within those workspaces, they can string together an array of analysis tools, either from a library of available tools or analysis tools of their own design. 5 minutes. The Kids First data resource is also working to make the data accessible to biomedical researchers with less extensive bioinformatics expertise. And a key part of this effort is the development of this variant explorer. It allows users to search, analyze, and visualize genomic variants using summary data from the Kids First data resource. I think this is extremely important because it allows genetic variants identified in Kids First participants to be filtered by variant type, gene frequency, pathogenicity, other factors. And it allows a bench scientist who isn't a bioinformatics person to go in, look at their gene of interest, and before they then start conducting functional validation studies to see what these variants are really doing in an animal model or a cell-based system. So what has the Kids First program been able to accomplish in 10 years? This figure illustrates the anatomical systems included in the Kids First data sets. We, and by we I mean the many, many people across the globe really who participated in Kids First by providing cohorts, being part of our sequencing centers or part of the Data Resource Center at CHOP, as well as those of us at NIH. We've been able to assemble one of the largest collections of pediatric genomic data with 35 studies from some 27 participants publicly available. In an earlier slide, it showed that 78 studies had been supported by EXO-1s from Kids First. And what that really indicates is there's a lot more data yet to be made publicly available. So you can expect this data resource to grow. That's just 3 of the most recent available studies. And I'm trying to speed up here. Personally, especially as I retire, I find it very satisfying to see, very gratifying to see that the Kids First data seems to be getting used. People are interested in it, and one measure of that is they're applying for access through dbGaP. That seems to be increasing rapidly, and in just the last quarter, we had 131 new approved dbGaP access requests. And I want to point out that first, that was more than we had in all of 2023. And it's access for secondary use of the data. Recall the people who supplied the cohorts already have access to their data, and their groups are already working on their individual projects, but this is for secondary use, and I find that very gratifying. Of course, for academic researchers, publications are like the gold standard, but it takes a long time to do the work, get the analysis out, and get it published. But so far, at least 160 research articles have come out using Kids First data. So I've talked about the main focus of effort for Kids First, that of building a data resource of sequence and associated data. However, our mandate to supplement, not supplant, other NIH research efforts allowed us much more flexibility than in most NIH programs. I'm not going to talk about those efforts, but we have been able to pursue pilot programs, add additional data modalities, support interoperability efforts across other NIH data efforts, and support development of new ways for making clinical and phenotypic data available. If the program's extended, there are a number of exciting initiatives that have been conceived for Kids First. And these I presented at a NIH Council of Councils last January, and the videotape of that presentation is available. But what I was most excited about is the notion that we may be able to bring clinical and phenotypic data from the clinic, from individual patients in the clinic, and make that data available for researchers. Of course, I've retired, so I'm no longer in the room where these initiatives can be discussed, and thankfully I won't be part of the really substantial work it takes to bring an initiative to fruition and make it available for the public. But the Kids First programs always tried to be responsive to the needs of the research community. So that's not going to change with any change in leadership. So your input on what directions to pursue will always be considered and appreciated, I'm certain. Ultimately, the future of Kids First rests with the U.S. Congress. There are short-term funding, at least for this next year through August of 2025. That funds are already available. And both House and Senate versions of funding for NIH include appropriations for Kids First. So there'll probably be a follow-up year, but that's not certain. However, this is short-term funding and doesn't allow new initiatives to be pursued. But fortunately, there are two authorization bills to extend the program. A House version passed the House last March and was referred to the Senate. And the Senate version was placed on their legislative calendar in October a year ago, but has sat there since. My understanding, and Adam probably knows better, but my understanding is that negotiations are ongoing to reconcile differences between these two bills. As a federal employee, I was legally not allowed to say anything to get your input into Congress. I couldn't lobby. But now I can, and what I want to say to y'all is that if you think that this data resource and continuing to build this data resource is valuable, then you should do what you learned in high school civics class and pick up the phone and contact your congressional representatives and say, Kids First, the Gabriella Miller Kids First Research Act 2.0 needs to be passed and passed into law before January 3rd, when the Congress ends and all the work setting this up has to go again.